The SomaScope Smart is a smart and easy-to-handle instrument. It's specifically designed for the analysis of somatic cells in raw milk on a small scale. It's based on flow cytometer technology. Somatic cells are white blood cells, or the body's defense mechanism, whose primary functions are to eliminate infections and repair tissue damage. The number of these cells in the milk gives an indication about the health of the cow and the quality of the milk. How does it work? There is a sample preparation unit where milk and a staining solution are combined, after which they are transported to the optic unit for counting. For daily use, there are 4 liquids in the SomaScope Smart that are easily removed, cleaned and filled. Decon solution, 5% for cleaning purposes. Number 2 is a Triton solution for rinsing. Number 3, the DAPI staining solution, marks the DNA cells in the milk so they can be analyzed by the flow cytometer. A brown bottle is used to protect the staining solution from light. This also differentiates the bottle from others. The 4th bottle is also a Triton solution, necessary for the so-called sheath flow that is inherent to the flow cytometer principle. The pipettes are labeled to avoid mixing the fluids. The concept of the instrument is based on clean and fresh liquids every day. Therefore, the size of the bottles is based on the number of around 200 samples analyzed on a working day. The sample preparation unit consists of a syringe, 2 peristaltic pumps, and a mixing unit. In the optic unit, we find the flow cell, 2 photomultipliers, and a microscope. The stained cells are passed one by one through the flow cell. The light of the power LED combined with the staining solution give the cells a fluorescent color, so they can be detected by the 2 photomultipliers. The SomaScope Smart uses 2 photomultipliers to better separate the cell signals from signals of other material like bacteria. The SomaScope Smart software is known for its user-friendliness. When starting up, the only thing the user has to do is log in. The Startup Wizard will appear automatically. The Startup Wizard guides the user through a few basic steps, refilling the bottles, adjusting the cassettes, and starting the cleaning process. The cleaning process takes several minutes. After this, the instrument is ready to go. When the instrument is switched on, a red light appears. A clear overview of all command actions for analysis, cleaning and calibration are shown on screen. The actual graphics of the somatic cell count measurements are displayed on the right. In the graphics, you see the results of photomultipliers 1 and 2 and a combined overview in order to compare the differences. The software also includes checking and adjusting areas of the hardware. Another useful tool is the GLP log. This stands for Good Laboratory Practice, and it enables the user to track all the previous actions. There is a possibility of creating and setting up personal favorites for colors, column settings, and text settings. Results can easily be transported to Excel for further processing. For measuring a sample, a warmed-up sample needs to be presented to the pipette. Now, the user selects the Measure button in the software, or the red button on the right of the instrument. This opens a screen where, if you choose, you can enter the name or specific identification of the sample. When this is not chosen, the software automatically generates a sample name followed by a number. Only one measurement will be taken from the presented sample. When it's required to measure more replicates from the same sample, the Repeat button needs to be selected. Otherwise, the sample needs to be removed and a new sample can be presented. The results of the analysis are stored on a day-to-day database. This makes it easy to track the results at a later stage. The SomaScope Smart uses a so-called secondary method. Therefore, it needs to be calibrated against reference samples, which are analyzed using a primary method called direct microscopic somatic cell counts. The user starts the calibration by choosing the calibration wizard. The reference samples are measured. After analyzing them, the instrument needs to be cleaned. After this, the reference values are entered into the reference column. Delta also supplies reference materials for the SomaScope Smart. These are non-milk official samples.